Hello and welcome dear students. Today we are going to discuss about the topic assumptions of Hardy-Weinberg equilibrium. The main objectives of today's lecture are to know the basics of Hardy-Weinberg equilibrium, to discuss in detail about the assumptions of Hardy-Weinberg equilibrium, to discuss about the role of mutations in Hardy-Weinberg equilibrium, to discuss about genetic drift in Hardy-Weinberg equilibrium, to discuss about mating patterns in Hardy-Weinberg equilibrium, and finally to understand the significance of Hardy-Weinberg law. Dear students, before going to the topic, let's understand certain key points regarding the topic. Godfrey Hardy and Wilhelm Weinberg postulated the Hardy-Weinberg principle in the early 20th century. It predicts both allele and genotype frequencies in populations which are non-evolving ones. The first condition that must be met for Hardy-Weinberg equilibrium is the lack of mutations in a population. The second condition that must be met for Hardy-Weinberg equilibrium is no gene flow in a population. The third condition that must be met is the population size must be sufficient so that there is no genetic drift. The fourth condition that must be met is random mating within the population. Finally, the fifth condition necessitates that natural selection must not occur. If the assumptions are not met for a gene, the population may evolve for that gene. That is, the gene's allele frequencies may change. Dear students, let's start with the introduction first. One of the most important principles of the study of genetic composition and differences in populations is the Hardy-Weinberg equilibrium principle, also described as Hardy-Weinberg equilibrium or Hardy-Weinberg law. This principle gives the genetic parameters for a population that is not evolving. In such a population, genetic variation and natural selection do not occur and the population does not experience change in genotype and allele frequencies from generation to generation. This law describes how a population can remain at genetic equilibrium, referred to as the Hardy-Weinberg equilibrium. Genetic equilibrium occurs when there is no evolution within the population. In other words, the frequency of alleles, variants of a gene will be the same from one generation to another one. At genetic equilibrium, the gene or gene frequencies are stable, they do not change. For example, let's assume that red hair is determined by the inheritance of a gene with two alleles R, that is capital R and small r. The dominant allele R encodes for non-red hair while the recessive allele small r encodes for red hair. If a population's gene pool contains 90% capital R and 10% small r alleles, then the next generation would also have 90% capital R and 10% small r alleles. However, this only works under a strict set of conditions. Dear students, let's discuss the assumptions of Hardy-Weinberg principle. The Hardy-Weinberg principle was developed by the mathematician Godfrey Hardy and physician Wilhelm Weinberg in the early 1900s. They constructed a model for predicting genotype and allele frequencies in a non-evolving population. This model is based on 5 main assumptions or conditions that must be met in order for a population to exist in genetic equilibrium. These 5 main conditions are like mutations must not occur to introduce new alleles to the population, no gene flow can occur to increase variability in the gene pool, a very large population size is required to ensure allele frequency is not changed through genetic drift, mating must be random in the population, natural selection must not occur to alter gene frequencies. The conditions required for genetic equilibrium are idealized as we don't see them occurring all at once in nature. As such, evolution does happen in populations. Based on the idealized conditions, Hardy and Weinberg developed an equation for predicting genetic outcomes in a non-evolving population over time. This equation, p square plus 2pq plus q square is equal to 1, is also known as the Hardy-Weinberg equilibrium equation. It is useful for comparing change in genotype frequencies in a population with the expected outcomes of a population at genetic equilibrium. In this equation, p square represents the predicted frequency of homozygous dominant individuals in a population, and 2pq represents the predicted frequency of heterozygous individuals, and q square represents the predicted frequency of homozygous recessive individuals. In the development of this equation, Hardy and Weinberg extended established Mendelian genetics principles of inheritance to population genetics. Let's study the assumption 1. Mutations don't happen. Mutations are errors that happen when DNA is being copied. That can be nonsense or neutral, that can be harmful to the organism, or that can change the organism in a way that makes them better able to survive and reproduce. Hardy-Weinberg equilibrium assumes that mutations don't exist because they represent genetic change that's not due to the recombination of existing alleles. One of the conditions that must be met for Hardy-Weinberg equilibrium is the absence of mutations in a population. Mutations are permanent changes in the gene sequence of DNA. These changes alter genes and alleles, leading to genetic variation in a population. Although mutations produce change in the genotype of a population, they may or may not produce observable or phenotypic change. Mutations may impact individual genes or entire chromosomes. Gene mutations typically occur as either point mutations or base pair insertions or deletions. In a point mutation, a single nucleotide base is changed, altering the gene sequence. Base pair insertions or deletions cause frame shift mutations in which the frame from which DNA is read during protein synthesis is shifted. This results in the production of a faulty protein. These mutations are passed on to subsequent generations through DNA replication. Although mutation is the original source of all genetic variation, mutation rate for most organisms is pretty low. So the impact of brand new mutations on allele frequencies from one generation to the next generation is usually not large. However, natural selection acting on the results of the mutation can be a powerful mechanism for evolution. Chromosome mutations may alter the structure of a chromosome or the number of chromosomes in a cell. Structural chromosome changes occur as a result of duplications or chromosome breakage. Should a piece of DNA become separated from a chromosome, it may relocate to a new position or another chromosome. That is the translocation. It may reverse and be inserted back into the chromosome, that is inversion, or it may become lost during cell division, that is called deletion. These structural mutations change gene sequences on chromosomal DNA, producing gene variation. Chromosome mutations also occur due to change in chromosome number. This commonly results from chromosome breakage or from the failure of chromosomes to separate correctly, that is non-disjunction, during meiosis or mitosis. Dear students, let's study the assumption number 2, that is no genetic drift. Sexual reproduction recombines genetic information in random pattern. In small population, it is possible that few individuals carry an allele and simple chance could make them more or less successful in passing on that allele. This is known as genetic drift, and the Hardy-Weinberg assumes that it does not happen. In practical terms, a population at Hardy-Weinberg equilibrium has to be large enough that the frequency of an allele is not impacted by random events. A very large population, one of infinite size, is required for Hardy-Weinberg equilibrium. This condition is needed in order to combat the impact of genetic drift. Genetic drift is described as a change in the allele frequencies of a population that occurs by chance and not by natural selection. Genetic drift involves change in allele frequency due to chance results or chance events, literally sampling error in selecting alleles for the next generation. Drift can occur in any population of non-infinite size, but has a stronger effect on small populations. The smaller the population, the greater the impact of genetic drift. This is because the smaller population, the more likely that some alleles will become fixed and others will become extinct. Removal of alleles from a population changes allele frequencies in the population. Allele frequencies are more likely to be maintained in large populations due to the occurrence of alleles in a large number of individuals in the population. Genetic drift does not result from adaptation but occurs by chance. The alleles that persist in the population may be either helpful or harmful to the organisms in the population. Two types of events promote genetic drift and extremely lower genetic diversity within a population. The first type of event is known as population bottleneck. Bottleneck populations result from a population crash that occurs due to some type of catastrophic event that wipes out the majority of the population. The surviving population has limited diversity of alleles and a reduced gene pool from which to draw. A second example of genetic drift is observed in what is known as the founder effect. In this instance, a small group of individuals become separated from the main population and establish a new population somewhere else. This colonial group does not have the full allele representation of the original group and will have different allele frequencies in the comparatively smaller gene pool. Dear students, let's discuss the assumption third. A closed population, that is, no gene flow will occur in that closed population. Immigration and immigration, that is, transfer of individuals into and out of the population, can change the frequency of alleles. Immigrating individuals might take more of one allele out of a population, and immigrating individuals might come from a population with a different proportion of alleles. This is known as genetic flow, and it is common for most populations, with notable exceptions such as remote islands, deep caves, or mountain tops. Gene flow involves the movement of genes into or out of population due to either the movement of individual organisms or their gametes. For example, eggs and sperms, like through pollen dispersal by a plant. Organisms and gametes that enter a population may have new alleles or may bring in existing alleles, but in different proportions than those already in the population. Gene flow can be a strong agent of evolution. A population at Hardy-Weinberg equilibrium is assumed to have no genetic flow or to be a completely closed population. At Hardy-Weinberg equilibrium, gene flow must not occur in the population. Gene flow or gene migration occurs when allele frequencies in a population change as organisms migrate into or out of the population. Migration from one population to another introduces new alleles into an existing gene pool through sexual reproduction between members of the two populations. Gene flow is dependent upon migration between separated populations. Organisms must be able to travel long distances or traverse barriers, that is, mountains, oceans, etc., to migrate to another location and introduce new genes into an existing population. In non-mobile plant populations, such as angiosperms, gene flow may occur as pollen is carried by wind or by animals to distant locations. organisms migrating out of the population can also alter gene frequencies. Removal of genes from the gene pool reduces the occurrence of specific alleles and alters their frequency in gene pool. Immigration brings genetic variation into a population and may help the population to adapt to environmental change. However, immigration also makes it more difficult for optimal adaptation to occur in a stable environment. The immigration of genes, that is, gene flow out of a population, could enable adaptation to a local environment but could also lead to the loss of genetic diversity and possible extinction. Dear students, let's now discuss the assumption 4, that is, random mating patterns. The Hardy-Weinberg principle assumes that every individual in a population has an equal chance of mating with every other individual, totally random mating. In a population at Hardy-Weinberg equilibrium, nobody gets to be picky. Random mating, also known as panmictic population, almost never happens in real life. Random mating is a condition required for Hardy-Weinberg equilibrium in a population. In random mating, individuals mate without preferences for selected characteristics in their potential mate. In order to maintain genetic equilibrium, this mating must also result in the production of the same number of offsprings for all females in the population. Non-random mating is commonly observed in nature through sexual selection. In sexual selection, an individual chooses a mate based on traits that are considered to be preferable. Traits such as brightly colored feathers, brute strength, or large antlers indicate higher fitness. In non-random mating, organisms may prefer to mate with others of the same genotype or of different genotypes. So non-random mating won't make allele frequencies in the population change by itself, though it can alter genotype frequencies. Females, more so than males, are selective when choosing mates in order to improve the chances of survival for their young. Non-random mating changes allele frequencies in a population as individuals with desired traits are selected for mating more often than those without these traits or characters. In some species, only select individuals get to mate over generations. Alleles of the selected individuals will occur more often in the population's gene pool. As such, sexual selection contributes to population evolution. Dear students, let's now discuss the assumption 5, that is, no natural selection. Natural selection is a pressure that favors one allele over another because the resulting trait or phenotype gives the individual an advantage. From a genetic standpoint, the only advantage that matters is one that helps the individual survive or reproduce more effectively in that population. In a population at Hardy-Weinberg equilibrium, there are assumed to be no evolutionary pressures. In order for a population to exist in Hardy-Weinberg equilibrium, natural selection must not occur. Natural selection is an important factor in biological evolution. When natural selection occurs, individuals in a population that are best adapted to their environment survive and produce more offspring than individuals that are not as well adapted. This results in a change in the genetic makeup of a population as more favorable alleles are passed on to the population as a whole. Natural selection changes the allele frequencies in a population. This change is not due to chance, as in the case with genetic drift, but the result of environmental adaptation. Natural selection occurs when one allele or combination of alleles of different genes makes an organism more or less fit, that is able to survive and reproduce in a given environment. If an allele reduces fitness, its frequency will tend to drop from one generation to the next generation. The environment establishes which genetic variations are more favorable. These variations occur as a result of several factors: gene mutation, gene flow, and genetic recombination during sexual reproduction are all factors that introduce variations and new gene combinations into a population. Traits favored by natural selection may be determined by a single gene or by many genes, that is, polygenic traits. Examples of naturally selected traits include leaf modification in carnivorous plants, leaf resemblance in animals, and adaptive behavior defense mechanisms such as playing dead. All these factors contribute to the change in gene frequency of a species in an area. If a few individuals from a species migrate to another place, the gene frequency changes again. It decreases from the place from where the individuals migrate and increases in the place they migrate to. If the frequency of the genes is high enough in the newly migrated land to start a new species, the migrated individuals become the founder species, and the effect is called founder effect. Thus, all these mechanisms contribute to the process of evolution. So, Hardy-Weinberg equilibrium never occurs in nature because there is always at least one rule being violated. Hardy-Weinberg equilibrium is an ideal state that provides a baseline against which scientists measure gene evolution in a given population. So, the Hardy-Weinberg equations can be used for any population. The population does not need to be in equilibrium. Dear students, let's now discuss the significance of Hardy-Weinberg law. The significance of Hardy-Weinberg law is that this model helps to evaluate the real genetic structure of a population over time. Only with the genetic composition we would predict if the population was in equilibrium with Hardy-Weinberg, that is, not evolving. In evaluating the genetic variation present in a population, Hardy-Weinberg law is essential and compares the real variation to the calculated value of Hardy-Weinberg law if the population was in equilibrium. If the real frequency in a population varies from the predicted value, then it is an indicator of one or more assumptions being disrupted and violated, which can be further investigated. It also helps in predicting the prevalence of a negative recessive gene in the heterozygous carriers. Dear students, let's now conclude the lecture. The Hardy-Weinberg model states that a population will remain at genetic equilibrium as long as 5 conditions are met, that is, 1. No change in the DNA sequence, no migration, a very large population size, random mating, and no natural selection. These 5 conditions rarely occur in nature. When one or more of these conditions does not exist, then evolution can occur. As a result, allele frequencies are constantly changing and populations are constantly evolving. As mutations and natural selection occur frequently in nature, it is difficult for a population to be at genetic equilibrium. The Hardy-Weinberg model also serves as mathematical formula used to predict allele frequencies in population at genetic equilibrium. If you know the allele frequencies of one generation, you can use this formula to predict the next generation. Again, this only works if all 5 conditions are being met in a population. Dear students, it was all about today's lecture regarding assumptions of Hardy-Weinberg equilibrium. Hope you have understood it well. See you next time with a different topic. Till then, take care. Goodbye.